So the paper that we shall be attempting today is May-June 2024, paper 2, variant 3. The first question states, the Zika virus is a pathogen that can infect human cells. Figure 1.1 is a drawing of the structure of a Zika virus. In figure 1.1, structure A is made of protein named structure A. So structure A is capsid, or you can write the protein code, but it is suggested that you write capsid because that is the name of the protein code, particularly. In figure 1.1, structure B is a single-stranded molecule, suggests the name of structure B. So it could be RNA as well as डीएनए because हमारे पास सिंगल स्ट्रैंडेड डीएनए भी वायरसेस में एग्जिस्ट करता है but single stranded RNA is also correct, so we would write RNA. Figure 1.2 is a transmission electron micrograph of a human kidney cells infected with Zika viruses. So they're showing us that these structures are basically Zika viruses within the kidney cells. Calculate the actual diameter of a Zika virus using the line XY. So this is the line XY which they've given us. In figure 1.2, show your working, give your answer in nanometer. So basically you will place a ruler against the line XY. The image size would come out to be 10 millimeters. Now we have to give our answers in nanometers. हम पूरी working कर सकते हैं और अपना n में answer nanometers में convert कर देंगे या फिर आप image size के answer को जो आपने image size की value को जो आपने 10 millimeters measure किया है उसको भी convert कर सकते हो nanometers में. So it's up to you if you want to do the conversion initially or afterwards. So the formula is magnification equals to image size upon actual size. Magnification is provided to us, which is 200,000 times 200,000. Image size is 10 millimeters. We have to find out the actual size. Now, हमारी जो ये value आएगी, it would be 10 divided by 200,000. This comes out to be 0.1234. Now, this value is in millimeters. We have to convert our actual size value to nanometers. So, millimeters to nanometer conversion के लिए millimeter to micrometer is times 1000. And from micrometer to nanometer is another times 1000. So, हमें basically अपनी millimeter वाली value को 10 to the power 6 से multiply करना होगा, and this would come out to be 50 nanometers. A magnification of times 200,000 cannot be achieved by a light microscope. The resolution of a transmission electron microscope is also higher than a light microscope. Describe what is meant by the resolution of a microscope. Resolution is basically the ability to distinguish between two things, or the minimum distance required between 2 points for them to be classified as distinct points. So, ability to distinguish between 2 points, or we can say the minimum distance needed between 2 points for them to be distinguished as two separate points. Both are acceptable. The vector for Zika virus is the mosquito Aedes aegypti. The mosquito feeds on the blood of an infected person and transmits the virus to another person when it feeds again. So the mosquito is acting as a vector, and through the blood meal of an infected person, it ingests the Zika virus, and if it takes the blood meal from an uninfected person, it would transfer the pathogen to the uninfected person while taking the blood meal. Describe the similarities and differences between the transmission of Zika virus disease and the transmission of malaria. So, for similarity, we would say that the transmission of malaria also is via mosquito vectors, And उन दोनों में यही होता है that both the vectors feed on blood, and whilst taking the blood meal, they transmit the disease from an infected person to an uninfected person. Difference क्या होगा that the vector, the mosquito which transfers the pathogen in malaria, is female Anopheles mosquito, and in Zika virus it is the Aedes aegypti mosquito. So हमारे पास जो vector की species है that is different, the mosquito species is different, which is responsible for transmission. Both the diseases require, both the diseases are transmitted. You can say both are blood-borne diseases. The vector which transmits plasmodium is female Anopheles mosquito, which is different from Aedes aegypti, which transmits Zika virus. Zika virus vaccine have been developed by scientists to explain how giving this vaccine to a person can lead to the development of long-term immunity against Zika virus disease. So the Zika virus vaccine would contain the Zika virus antigen, which would trigger an immune response. So vaccine contains Zika virus antigen, which would trigger an immune response, resulting in the production of antibodies and memory B cells complementary to the antigens of Zika virus. The memory B cells stay in the blood for a long time duration and would produce a faster secondary immune response if exposed again, if the person is exposed again to Zika virus. Explain how a vaccination program may limit the spread of Zika virus disease through a population. So when majority of the population is vaccinated, हमारे पास क्या कहते हैं, herd immunity develop हो जाती है, which basically provides an indirect protection to the population which is uninfected. तो हमारे पास क्या कहते हैं, when a large number of people are vaccinated against a particular pathogen, it basically decreases the prevalence of the pathogen in the population, and even the unvaccinated people are less likely to develop that particular disease. Herd immunity is developed through vaccination program when large proportion of the population is vaccinated It provides an indirect protection to the unvaccinated people, as they too are less likely to develop that disease. Clear? Any questions about question number one? Okay. Collagen is a fibrous protein that is found in many tissues in animals. Describe the structure of a collagen molecule and the structure of a collagen fiber. Okay, so in collagen molecule basically हमारे पास ट्रोपोकोलाजन molecule को हम बुलाते हैं. Now हमारे पास जो ट्रोपोकोलाजन होता है, ट्रोपोकोलाजन basically consists of three helical chains, which are basically consisting of glycine as every third amino acid in the three helical chains is glycine, and they associate via hydrogen bonds to form the collagen molecule, जिसको हम tropocollagen बुलाते हैं. So we'll write that down. So a collagen molecule is made up of three helical polypeptide chains. Troponin, yeah, that one. I'm wondering it must be very hard to actually keep track of all 19 chapters. I can't even imagine being a composite student ever. Breaking A levels into two parts is a very wise thing Cambridge people did. Collagen molecule is made up of three helical polypeptides, in which every third amino acid is glycine. The three chains tightly wound around each other via hydrogen bonds. Okay, for that, let me take you guys. हमारे पास basically बहुत सारे tropocollagen molecules associate via covalent crosslinks to form fibrils और फिर वो fibrils collagen fiber बनाते हैं. Let me show you guys the structural organization. So let's talk about collagen. Collagen में हमारे पास basically पहले collagen molecule बनता है जिसको हम tropocollagen बुलाते हैं, which basically is a tight coil of three helical polypeptide chains in which every third amino acid is glycine. हमारे पास क्या कहते हैं, जो ये ट्रोपोकोलाजन मॉलिक्यूल होता है, इट देन एसोसिएट्स टू फॉर्म कोलेजन फिब्रिल्स. कोलेजन अच्छा ये किस तरह से एसोसिएट करता है वाया कोवलेंट क्रॉस लिंक्स. सो हमारे पास क्या कहते हैं, न्यूमरस ट्रोपोकोलाजन मॉलिक्यूल्स जॉइन टुगेदर, दे आर लाइंग पैरेलल टू वन अनदर. आप देख सकते हो एक ट्रोपोकोलाजन दूसरा ट्रोपोकोलाजन, दे आर यूजुअली पैरेलल टू वन अनदर and they associate via covalent crosslinks, covalent bonds के through, covalent crosslinks के through, they associate to form a collagen fibril. This structure is a collagen fibril, and then numerous collagen fibrils collectively form a collagen fiber. Is this clear? Okay. So the collagen molecules are arranged in parallel. They associate via covalent crosslinks to form a collagen fibrils. Okay, there's a lag. Wait a second, let me change my location. Okay. A collagen molecule और fiber का difference समझ आ गया, and I hope अब screen lag नहीं करेगी ना तो expert होगी. Some amino acids in collagen can be modified to improve the stability of the protein. For example, the amino acid lysine can be modified to form hydroxylysine. Figure 2.1 shows a disaccharide bonded to the amino acid. So there's a disaccharide which is bonded to the amino acid hydroxylysine in a collagen molecule. The disaccharide is made from two monosaccharides, which are indicated by the labels D and E. So basically, this is the disaccharide, and हमारे पास क्या कहती हैं, this is the central carbon. कि हम आइडेंटिफाई करें डिफरेंट पार्ट्स को. हमारा ये सेंट्रल कार्बन है. दिस सेंट्रल कार्बन इज कनेक्टेड टू हाइड्रोजन एटम अमीन ग्रुप. ये अमीन ग्रुप क्या कहते हैं, एक पेप्टाइड बॉन्ड फॉर्म कर रहा है, तो इसने एक एज जो है अपना लूज कर दिया. सो ये हमारे पास बेसिकली इस तरह से बॉन्डेड होगा. यहाँ पर ये वाला पार्ट मिसिंग है, यहाँ पर एन और एच वाला पार्ट मिसिंग है. एंड दिस इज बेसिकली द कार्बोक्सिल पार्ट. और ये हमारे पास जो पूरा है, दिस इज बेसिकली द R group of the amino acid. ठीक है. अच्छा जी. Anybody who's confused about the labels? Okay. Give the precise name of the monosaccharide labeled D in figure 2.1. हमें D को identify करना है. अच्छा जी, हमारे पास this is basically a hexose sugar. और हमारे पास क्या कहते हैं, H is pointing upwards. नीचे हमारे पास OH होगा. उस OH का जो H है वो basically यहाँ पर bond form कर रहा है. So because the OH is pointing downwards, this would have been the alpha glucose. Very good. On figure 2.1, label the glycosidic bond with the letter G. Glycosidic bond वो होता है जो कि form होता है between carbon of one sugar, carbon of another sugar, and then there is an OH between. So हमारे पास this is basically the glycosidic bond. On figure 2.1, draw a circle around the R group of hydroxylysine and label the circle with the letter R. वो हम पहले ही कर चुके हैं कि ये हमारे पास हमारा R group है. So NH2 से लेकर पूरा CH2 is your R group. हम O और H वाले पॉइंट को नहीं लेंगे. O ये बेसिकली हमारे पास वो बॉन्ड है जो कि हमारे डाइसैक्राइड को कनेक्ट करे R ग्रुप से. सो हमारे पास दिस इज बेसिकली द एसोसिएटिंग लिंक बिटवीन द डाइसैक्राइड एंड द R ग्रुप ऑफ द अमीनो एसिड. सो ये हमारे पास ये पूरी चीज R ग्रुप का पार्ट नहीं है, बिकॉज हमें बताया हुआ है दैट सम अमीनो एसिड्स इन कोलेजन कैन बी मॉडिफाइड टू इम्प्रूव द स्टेबिलिटी ऑफ द प्रोटीन. For example, the amino acid lysine can be modified to form hydroxylysine. And they've told us that the figure shows a disaccharide bonded to the amino acid hydroxylysine in a collagen molecule. तो हमारे पास ये जो है ये R ग्रुप का पार्ट नहीं है, ये मॉडिफाइड वर्जन ऑफ द अमीनो एसिड है. ठीक है. Osteogenesis imperfecta. Osteogenesis imperfecta is a disease that results from a deficiency in collagen. Suggest how a named tissue or structure is affected in a person who has this disease. Now tell me, हमने कहाँ collagen पढ़ा हुआ है? In our syllabus, where have we studied about collagen? Um, blood vessels, yes, because अनस muscles जो हैं वो हमने क्या कहते हैं AS में नहीं पढ़े, right? So blood vessels में हमने collagen पढ़ा हुआ है. And we've talked about collagen. बायोलॉजिकल मॉलिक्यूल्स में हमने बात की थी कि जो कोलेजन है वो हमारे पास बेसिकली बोन्स में प्रेजेंट होता है. It basically makes up the more majority part of our skeleton. तो हम bones या blood vessels के point of view से में से किसी एक की बात कर लेंगे. हमारे पास कोलेजन का काम होता है स्ट्रेंथ प्रोवाइड करना. तो हमारे पास क्या कहते हैं, अगर हम ब्लड वेसल्स की बात करें तो ब्लड वेसल्स में अगर कोलेजन प्रेजेंट नहीं होगा, लेट्स से आर्टरीज में ब्लड आर्टरीज में हमारे पास क्या कहते हैं, अगर कोलेजन प्रेजेंट नहीं होगा तो हमारे पास क्या कहते हैं, आर्टरीज जो हैं वो चेंजेस इन ब्लड प्रेशर नहीं क्या कहते हैं बेयर कर पाएंगे. ये गैस एक्सचेंज सर्फेसिस पे भी होता है. सो हमारे पास क्या कहते हैं, जिस भी टिश्यू की आप बात करो उस टिश्यू से क्या कहते हैं, My voice is lagging. Wait. ओके, ठीक है। सो बेसिकली किसी भी हम टिश्यू की बात करें, उस टिश्यू का हम नाम लेंगे जहाँ पर कोलेजन प्रेजेंट होता है, ब्लड वेसल्स, बोन, स्किन, जिनके भी आप लोगों ने नाम लिए। बेसिकली वो जो भी स्ट्रक्चर है वो वीकर हो जाएगा, उसकी स्ट्रेंथ लेस हो जाएगी। अगर फॉर एग्जांपल स्किन की किसी ने बात की, स्किन में हमारे पास ये कहते हैं स्किन की जो टाइटनेस है, स्किन की जो स्ट्रेंथनिंग है वो कम हो जाएगी एंड स्किन वुड बिकम की कहते हैं वीकन, रिड्यूस स्ट्रेंथ हो जाएगी। सिमिलरली विद बोन्स, अगर बोन्स की हम बात करें, बोन्स में अगर कोलेजन डेफिशिएंसी हो जाएगी सो बोन विल बिकम वीकर। और बोन्स में ये भी हो सकता है कि अगर बोन्स वीकर हो जाएंगी तो वो इजिली ब्रोकन हो जाएंगी। आर्टरीज की अगर हम बात करें तो हमारे पास क्या कहते हैं, आर्टरीज चेंजेस इन ब्लड प्रेशर ड्यू टू वेंट्रिकुलर कॉन्ट्रैक्शन नहीं बेयर कर पाएंगी। सो ईदर वेज किसी भी टिश्यू के बारे में हम लिख सकते हैं। I would have personally written about bones. So bones would become weaker and more vulnerable to fractures. Next question. Arteries, capillaries, and veins are three types of blood vessels. Table 3.1 shows some features of these three types of blood vessel. Complete table 3.1 by using a tick if the feature is present in the blood vessel and a cross if the feature is absent from the blood vessel. Put a tick or a cross in every box. Smooth muscles, endothelium, tunica, media. अरे, तो हमारे पास जो स्मूथ मसल्स होते हैं वो आर्टरीज में प्रेजेंट होते हैं, वेन्स में प्रेजेंट होते हैं। कैपिलरी इज ओनली मेड अप ऑफ एंडोथेलियल सेल्स, तो हमारे पास स्मूथ मसल्स नहीं होंगे बिकॉज स्मूथ मसल्स कहाँ पर प्रेजेंट होते हैं, ट्यूनिका मीडिया में प्रेजेंट होता है। स्मूथ मसल्स एंड इवन इलास्टिन फाइबर्स, तो हमारे पास स्मूथ मसल्स हो गए, इलास्टिन फाइबर्स हो गए, ट्यूनिका मीडिया में प्रेजेंट होते हैं, जो ऑब्वियसली कैपिलरी में नहीं होती है। तो हमारे पास स्मूथ मसल्स भी कैपिलरी में नहीं होंगे। एंडोथेलियल सेल्स हमारे पास तीनों में होंगे, ट्यूनिका मीडिया खाली आर्टरी और वेन में होती है। Because 3.1 shows a transmission electromicrograph of an alveolus and part of the adjacent capillary. So ये हमारे पास alveolar ear space है और हमारे पास ये capillary lumen है जिसमें हमारे पास blood present होगा। हमें कोई सेल भी नजर आ रहा है। Draw two labeled arrows on figure 3.1 to show the direction of movement of oxygen and direction of movement of carbon dioxide during gas exchange in the लंग्स। तो ऑब्वियसली हमारे पास क्या कहती हैं, जो ऑक्सीजन है वो एल्वियोलर एयर स्पेस से कैपिलरी में मूव कर रही होगी और जो हमारे पास कार्बन डाइऑक्साइड है दैट वुड बी लीविंग द कैपिलरी ल्यूमेन and moving towards alveolar airspace. Suggest and explain how a steep oxygen concentration gradient is maintained in the lungs. We've talked about it a multiple of times. कि हमारे पास क्या कहते हैं, जो हमारी कांस्टेंट एल्वुलर वेंटिलेशन होती है। कांस्टेंट एल्वुलर वेंटिलेशन का मतलब यह है कि हमारे पास इनहेलेशन एंड एक्सहेलेशन जो होती है। Guys, tell me, is my voice lagging again? Because मेरी bandwidth low आ रही है, connection bandwidth. Okay, there is no lag. ठीक है, सही है। हमारे पास क्या कहते हैं, when we talk about the gas exchange system, let's say हमारे पास alveoli है। This is our alveoli and this is the capillary. ना हमारे पास होता क्या है, हमारे पास constantly oxygen rich air is coming in and oxygen deficient air is going out. So the air rich in oxygen coming in, oxygen deficit air is moving out. हमारे पास constantly deoxygenated blood आ रहा होता है और oxygenated blood जा रहा होता है। तो हमारे पास इस तरह से एक continuous concentration gradient is maintained for gas exchange to take place. So we'll write this down that blood arriving in the capillaries from the pulmonary artery is deoxygenated, or you can say has a lower oxygen concentration. The inhalation of air brings oxygen rich air in the alveolar space. As soon as gas exchange occurs, the oxygenated blood leaves the capillary. The air, which has low partial pressure, or you can say low oxygen concentration, is exhaled out. We can also talk about that there is a large network of capillaries which is surrounding the alveoli, increasing the surface area for gas exchange to take place. And this way, the alveolar ventilation, the continuous blood flow, and the network of capillaries surrounding the alveoli collectively maintain a steep oxygen concentration gradient for gas exchange. Network capillaries surrounding the alveoli increase the surface area diffusion. The passage outlines the roles of blood vessels associated with the heart. Complete the passage by using the most appropriate scientific terms. The dash carries blood to the left atrium. Left atrium. Left atrium receives oxygenated blood. So the pulmonary vein carries blood to the left atrium. After passing from the left atrium to the left ventricle, blood is pumped into the aorta. The aorta is one of two large arteries that carry blood away from the ventricles of the heart. Blood that leaves the heart to enter these arteries must pass through the dash valve. So हमारे पास क्या कहते हैं? One of the two large arteries that carry blood away, one is aorta, the other artery which carries the blood away from the ventricles of the heart is the pulmonary artery. So the blood moving from the ventricles to either the pulmonary artery or the aorta has to pass through the semi-lunar valves. Oxygenated blood is supplied to the cardiac muscle cells through the dash arteries. Those are your coronary arteries. Clear? Any questions? Question number 4 states lignin and suberin are polymers that are present in plant tissues. Lignin हमारे पास कहाँ present होता है? It is present in the xylem vessel walls. So in the walls of the xylem vessels हमारे पास lignin present होता है. And हमारे पास suberin is basically present in the endodermal cells and forms the Casparian strip. Describe and explain the roles of lignin and suberin in the transport of water through the roots and stem of a plant. So basically, हमारे पास lignin and suberin ये दोनों hydrophobic in nature. And hydrophobic in nature means that they do not allow water to pass through them. So water cannot pass through lignin, water cannot pass through suberin. Now, the purpose of suberin is that it is present in the endodermis and it blocks the apoplast pathway and forces the water to move via the symplast pathway in order to ensure that no toxic substances, no toxic solutes are entering in the xylem vessel. So its purpose, suberin's purpose, is to block the apoplast pathway, cause all the water to pass through the symplast pathway so that everything which is present in the water, which is going to enter inside the xylem, is passing through a cell surface membrane so that only the substances which are necessary enter the xylem and any toxic substance, any pathogen, it is basically prevented from entering in the xylem vessel. When we talk about lignin, lignin is found in the walls of xylem. And in the walls of xylem, it provides structural support by preventing the collapse of xylem vessel due to high water pressure. And it also ensures कि हमारे पास जो water है it does not leak through the xylem vessel. So lignin and suberin are hydrophobic in nature. Suberin is found in the endodermal cells and it ensures that water moves through the symplast pathway in order to prevent the entry of toxins in the xylem. Lignin is found in the walls of xylem vessels where it provides mechanical support and prevents loss of water from xylem. The enzyme lactase catalyzes the formation of lignin in plants. Figure 4.1 is a diagram of the mode of action of lactase. So this is basically the active side of the lactase enzyme. We have copper ions. The copper ions would be acting as your cofactors. Now, the we can see that the the monolignols, which are the substrate of your enzyme lactase, they are not perfectly complementary. The active site and the shape of the substrate is not perfectly complementary. However, the in the second stage, we can see that the shape of the active site of the enzyme is changing according to the structure of the substrate. And once the substrate has left, as when the substrate has turned into a product, the and it has left the active site is back to its original shape. Now, this mode of action is your induced fit model. So we have to identify, describe and explain the mode of action of lactase when catalyzing the formation of lignin. So हमारे पास क्या कहते हैं? We'll first mention the name of the model, which is the induced fit model. Then we'll describe what happens in the induced fit model, allowing for the catalyzation of the lignin molecule. So the enzyme lactase uses the induced fit model initially or initially the active site of the enzyme is not completely complementary to the shape of substrate, which is monolignols. The active site changes its shape according to the shape of monolignals to become fully complementary enzyme substrate complex is formed once the product lignin chain has formed and left the active site. The enzyme returns to its original shape. Next question. Cyclin dependent kinases are enzymes that regulate the cell cycle. Cell signaling by ligands causes the activation of CDKs in target cells. So, हमारे पास these enzymes are basically activated as a response of a cell signaling molecule known as ligands. Outline the main stages in the process of cell signaling by ligands that can cause specific responses in target cells. So, हमारे पास क्या कहते हैं? The ligands are transported through the bloodstream and they bind to complementary receptors on the target cell, triggering a series of reactions to take place in the target cell. So ligands travel within the bloodstream and bind to complementary receptors on the target cell, resulting in a series of reactions to take place within the target cells, such as the activation of enzymes. The activity of CDKs are basically the enzymes, is reduced by CDK inhibitors. So these enzymes activity is reduced by their inhibitors. Many of these inhibitors occur naturally in the cells. So इनके cells के अंदर ही आपके CDK inhibitors पाए जाते हैं. नहीं, आपको इतनी ही इंफॉर्मेशन पता होनी चाहिए सेल सिग्नलिंग के बारे में दैट cell signaling molecules are produced from the site of production, from the site of origin, and they travel or are transported by the circulatory system in the bloodstream. They bind to complementary receptors on the target cell. Once they bind to the complementary receptors, they trigger enzyme cascade reactions or they trigger a series of reactions within the target cell. That's all you need to know about सेल सिग्नलिंग. हाँ, आपको सेल सिग्नलिंग मॉलिक्यूल्स का जो साइंटिफिक टर्म है लिगेंड वो साइंटिफिक टर्म भी पता होनी चाहिए. The activity of CDKs is reduced by CDK inhibitors. Many of these inhibitors occur naturally in cells. Figure 5.1 is a diagram of a CDK inhibitor binding to a CDK molecule. So, okay, so this is the inhibitor. The inhibitor is not binding to the active site. Instead, it is binding to the allostatic site, which means that it is a non-competitive inhibitor. State and explain how the CDK inhibitor in figures 5.1 prevents the activity of the CDK molecule. We would say that the inhibitor is a non-competitive inhibitor binding to the allosteric site of the enzyme, due to which the shape of the active site would change, resulting in the active site no longer being complementary to the substrate, thus preventing the formation of enzyme substrate complexes. The inhibitor is a non-competitive inhibitor binding to the allosteric site. This causes the shape of the active site to change, and thus the active site is no longer complementary to the substrate. Table 5.1 lists three different CDKs, their roles in the cell cycle, and molecules that inhibit them. So we have three CDK enzymes, CDK1, CDK2, CDK4 listed. CDK1 regulates cells progression from G2 to mitosis. So आपके पास जो second growth stage है, उससे माइटोसिस की स्टेज यानी कि प्रोफेज तक प्रोग्रेस करवाता ही है वो एंजाइम. इसका जो इनहिबिटर है दैट इज RO3306. CDK2 is important to regulate the processes of the S phase. S phase में हमारे पास DNA replication हो रही होती है. The inhibitor of this enzyme is P21 CIP1. CDK4 regulates cell progression from G1 to S phase, and palbociclib is the inhibitor of this particular CDK. With reference to table 5.1, state which CDK inhibitor is likely to result in a cell containing one chromatid per chromosome. One chromatid per chromosome का मतलब ये है that DNA replication has not occurred. Now tell me, कौन सा inhibitor ये कर सकता है, DNA replication को रोक सकता है? Which inhibitor's activity would cause in CDK1 is the name of the enzyme. We have to state the inhibitor. Inhibitor ये है. Which inhibitor would have an effect on the process of DNA replication? P21 CIP1. So हमारे पास हो सकता है that. अच्छा हमारे पास है P21 CIP1 नहीं, हमारे पास even palbociclib, इन दोनों में से कोई सी भी एक हमारे पास क्या कहते हैं ये कॉज कर सकती है. दोनों का नाम नहीं लिखना, किसी एक का नाम लिखना, but I'll explain why either of the two can result in the cell containing one chromatid per chromosome. So हमारे पास क्या कहते हैं, stages क्या होती हैं cell cycle की? We have G1. G1 के बाद S phase होता है. S phase में हमारे पास DNA replication होती है. S phase के बाद G2, और G2 के बाद we have mitosis, जिसमें हमारे पास PMAT आ जाता है, प्रोफेज, मेटाफेज, एनाफेज एंड टीलोफेज, एंड देन वी हैव साइटोकाइनेसिस. ये हमारे पास सेल साइकिल की स्टेजेस होती हैं. नाउ हमारे पास CDK2 एंजाइम का जो रोल है वो हमारे पास एस फेज में होता है. सो CDK2 एंजाइम इज इम्पोर्टेंट फॉर रेगुलेशन ऑफ द प्रोसेस ऑफ द एस फेज. सो DNA रेप्लिकेशन में इसका हो सकता है एक इम्पोर्टेंट रोल. सो इफ P21 CIP1 inhibitor basically is present or binds to CDK2 and prevents it from catalyzing the processes of the S phase, हमारे पास DNA replication would stop, and हमारे पास क्या कहते हैं that would result in the cell containing one chromatid per chromosome. ठीक है, हमारे पास normal DNA replication नहीं होगी. However, हमारे पास अगर हम बात करें palbociclib की, palbociclib is basically the inhibitor of the enzyme, which is important for transitioning the cell from G1 to S phase. अगर हमारे पास ये ट्रांजिशन ही रुक जाए, हमारे पास G1 के बाद जो सेल है वो बाकी नेक्स्ट स्टेज में प्रोग्रेस ना करे. So if the cell is not progressing from G1 to S phase, would DNA replication occur? If the cell never enters the S phase, हमारे पास क्या DNA replication होगी? या हमारे पास क्या कहते हैं, the roles which are mentioned, अगर inhibitor उन enzyme से bind कर देगा तो obviously those enzymes would not be able to carry out their roles, and therefore हमारे पास जो हमारी enzymes important है for the processes of S phase and the enzyme which is responsible for progressing the cell from G1 to S phase, इन दोनों में अगर क्या कहते हैं, इन दोनों की एक्टिविटी में hindrance आ जाए, inhibition आ जाए, तो हमारे पास क्या कहते हैं, DNA replication या तो बिल्कुल नहीं होगी. In this case, no DNA replication occurs if palbociclib inhibits CDK4, and if P21 CIP1 inhibits CDK2, हमारे पास hindrance और disruption आ जाएगी in the process of DNA replication. So इनमें से किसी एक एंजाइम की हम बात करेंगे. Yes, the RO3306 would not be causing the cell to contain one chromatid per chromosome, because इसका जो role है, ये जिस enzyme से bind करता है, उस enzyme का role is after DNA replication has taken place. वो इधर इस progression के लिए important होती है, and obviously till this point DNA replication हो चुकी भी होती है. ठीक है? Okay. With reference to table 5.1, state which CDK inhibitor is likely to result in a cell with a relatively high concentration of mitochondria, 2 chromatids per chromosome. तो हमारे पास क्या कहती हैं, 2 chromatids per chromosome. ये हमारे पास बेसिकली एक ऐसा सेल जिसमें 2 chromatids per chromosome है, इसका मतलब है उस सेल में DNA replication has occurred. Do we agree with this? DNA replication has occurred. तो हमारे पास DNA replication में कोई hindrance नहीं आई. इसका मतलब है कि ये P21 CIP1 और palbociclib inhibitors की वजह से तो नहीं हुआ, क्योंकि अगर P21 CIP1 होते या palbociclib होते तो हमारे पास 2 chromatids per chromosome present नहीं होता. So, with reference to table 5.1, state which CDK inhibitor is likely to result in a cell with 2 chromatids per chromosome. ये तो हमें समझ आ गया कि हमारे पास दो क्रोमेटिड्स पर क्रोमोसोम तभी होंगे जब हमारे पास डीएनए रेप्लिकेशन इनहिबिट नहीं होगी. सो हमारे पास दिस इज आउट एंड दिस इज आउट. सो ऑब्वियसली RO3306 अगर इनहिबिट कर रहा है तो हमारे पास उसकी इनहिबिशन की वजह से डीएनए रेप्लिकेशन पर कोई फर्क नहीं पड़ेगा, एंड देयरफॉर वी वुड बी हैविंग 2 chromatids per chromosome. The question is, why is there a relatively high concentration of mitochondria? Now, हमारे पास क्या कहते हैं, अगर हमारे पास RO3306 इस पॉइंट पर एक्ट करता है, इज दैट करेक्ट? G2 से माइटोसिस की प्रोग्रेशन वाली एंजाइम जो है, उस एंजाइम की एक्टिविटी को इनहिबिट करता है. सो दैट मीन्स कि माइटोसिस नहीं होगा और साइटोकाइनेसिस नहीं होगा इफ द इनहिबिटर RO3306 इज प्रेजेंट. राइट, तो हमारे पास द सेल स्टेज इन G2. The cell, because of the presence of RO3306, stays in the G2. G2 का क्या मतलब होता है? ग्रोथ फेज 2. हमारे पास जो ग्रोथ फेज होती है, ग्रोथ फेज में आपके पास क्या कहते हैं, ऑर्गेनील सिंथेसिस हो रहा होता है, प्रोटीन सिंथेसिस, लिपिड सिंथेसिस, सारे सिंथेसिस आपके पास ड्यूरिंग योर इंटरफेस हो रहे होते हैं. तो जो आपका पूरा इंटरफेस का टाइम है G1 to G2, आपके पास ऑर्गेनील्स के नंबर्स बढ़ रहे होते हैं, मॉलिक्यूल्स बन रहे होते हैं. इज दैट करेक्ट? नाउ हमारे पास अगर सेल अरेस्टेड है ग्रोथ फेज 2 में, तो हमारे पास ऑर्गेनील सिंथेसिस हो रहा होगा. माइटोकॉन्ड्रिया इज एन ऑर्गेनील, इट इज अ सेल्फ रेप्लिकेटिंग ऑर्गेनील, इट कैन इंक्रीज इट्स नंबर. सो हमारे पास ग्रोथ फेज 2 में माइटोकॉन्ड्रिया का सिंथेसिस हो रहा होगा, एंड अगर माइटोकॉन्ड्रिया का सिंथेसिस हो रहा है, माइटोकॉन्ड्रिया की सेल्फ रेप्लिकेशन हो रही है, तो हमारे पास माइटोकॉन्ड्रिया का नंबर इंक्रीज हो रहा है. साइटोकाइनेसिस में क्या होता है? साइटोकाइनेसिस में जो भी ऑर्गेनील्स होते हैं, जो भी चीजें साइटोप्लाज्म में प्रेजेंट होती हैं, दे आर डिवाइडेड बिटवीन टू सेल्स। हमारे पास साइटोकाइनेसिस ही नहीं हो रहा है, क्योंकि अगर प्रीमैटरी स्टेजेस तक हमारी प्रोग्रेशन नहीं हो रही तो साइटोकाइनेसिस भी नहीं हो रहा है। इसका मतलब ये है कि जो भी ऑर्गेनील्स हैं, दोज ऑर्गेनील्स आर स्टेइंग विद इन दिस सेल, अरेस्टेड इन द G2 phase, and they are not being divided, and therefore we are seeing a high concentration of mitochondria in the cell. So RO 3306, the explanation would be the cell cycle is arrested in G2 phase. Thus, mitochondria are increasing in number due to organelle synthesis. And since cytokinesis is not occurring, their numbers the numbers of mitochondria remain high. आप या तो माइटोकॉन्ड्रिया की रिस्पेक्ट से एक्सप्लेनेशन लिख सकते थे, या फिर आप टू क्रोमैटिड स्क्वायर क्रोमोसोम के रिस्पेक्ट से भी एक्सप्लेनेशन लिख सकते थे, दैट डीएनए replication occurs before G2 phase in the S phase. तो आप किसी भी एक पॉइंट के एस्पेक्ट से एक्सप्लेनेशन लिख सकते थे. Scientists have developed CDK inhibitors that are synthetic. Explain why CDK inhibitors can be used to treat cancerous tumors. हमारे पास cancerous tumors का uncontrolled डिविजन होती है, अनकंट्रोल्ड माइटोसिस हो रहा होता है. अगर हम सीडीके इनहिबिटर्स यूज करेंगे, सो वी कैन सी कि हमारे पास द सीडीके इनहिबिटर्स वुड प्रिवेंट द माइटोसिस अकरिंग, क्योंकि हमारे पास अगर लेट्स से एस फेज नहीं हो रही, या हमारे पास जो सेल है वो G2 में अरेस्ट हो जाएगा, सो वो माइटोसिस तक प्रोग्रेस नहीं कर पाएगा. अगर माइटोसिस तक प्रोग्रेस नहीं कर पाएगा तो हमारे पास अनकंट्रोल्ड सेल डिविजन रुक जाएगी. So, cancerous tumors develop due to uncontrolled mitosis. CDK inhibitors stop the cell cycle before mitosis, thus preventing uncontrolled cell division. Next question. DNA and RNA are polynucleotides. Describe 3 ways in which the structure of mRNA differs from the structure of DNA. In each of your answer, include information about the structure of mRNA and the structure of DNA. तो हमने कंपैरिजन में दोनों की बात करनी है कि mRNA की कौन सी चीज डिफरेंट है, DNA की कौन सी चीज. जैसे हमारे पास we'd say that mRNA is a single-stranded molecule, whereas DNA is a double-stranded molecule. mRNA is a single stranded molecule, whereas DNA is a double stranded molecule. Then हमारे पास क्या कहते हैं, ribose sugar is present in RNA nucleotides, whereas deoxyribose sugar is present in DNA nucleotides. Ribose sugar is present in RNA nucleotides, whereas deoxyribose sugar is present in DNA nucleotides. फिर हमारे पास क्या कहते हैं? Uracil is found alongside adenine, cytosine, guanine in RNA, whereas thymine is found in DNA alongside adenine, cytosine, and guanine. The bases found in RNA include adenine, cytosine, guanine, and uracil, whereas the bases found in DNA are adenine, guanine, cytosine, and thymine. Scientists have synthesized four synthetic bases Z, P, S, and B. The base pairings of the synthetic bases are shown in figure 6.1. Synthetic का मतलब है artificial है. They're not organic or naturally found bases. ये laboratory में synthetically बनाई गई हैं. So एक हमारे पास ये base है. Or it's a Z. This is basically P. Then we have S and we have B. These are the four bases which have been formed by the scientist. D is the site where bond forms with deoxyribose. So यहाँ पर हमारे पास शुगर होगी. यहाँ पर शुगर है, यहाँ पर शुगर है, यहाँ पर शुगर है. State the letters that represent two purine synthetic bases. हमारे पास purine कौन सी bases होती हैं? Double ring bases होती हैं. So double rings किन में है? हमारे पास P में हमें double ring दिखाई दे रही है. This is double ring. और हमें B में डबल रिंग दिखाई दे रही है. So this is a double ring base. So P and B are acting like purines. State and explain which DNA base pair. Yes, Dua. Have I missed something? Yes, sure. You can discuss the questions. We're done with this paper. इसके बाद आप पूछ सकते हो. State and explain which DNA base pair is most similar to the synthetic base pairs in figure 6.1. अच्छा जी. Now मैं इसको erase कर रही हूँ. हमारे पास एक बेस ये है P और एक बेस ये है Z, एक बेस B, एक बेस S. हमारे पास कितने हाइड्रोजन बॉन्ड्स हमें नजर आ रहे हैं बिटवीन दीज बेसेस? इन दोनों के बीच में कितने हाइड्रोजन बॉन्ड्स हैं? Z और P के बीच में, और S एंड B के बीच में कितने हाइड्रोजन बॉन्ड्स हैं? 3 हाइड्रोजन बॉन्ड्स, 3 हाइड्रोजन बॉन्ड्स. नेचुरल सर्कमस्टांसेस में हमारे पास कौन सी दो DNA बेसिस होती हैं जिनके दरमियान 3 हाइड्रोजन बॉन्ड्स बनते हैं? Which two bases associate with 3 hydrogen bonds? Adenine and thymine associate via 2 hydrogen bonds, whereas cytosine and guanine associate via 3 hydrogen bonds. So, cytosine and guanine, since they associate via 3 hydrogen bonds, similar to the synthetic bases shown. And with that, we are done with this paper.